This is the process of protein synthesis. The purpose of this process is to use a code from a DNA strand, which is the order of the base pairs, and to create a protein. There are two steps to this process: transcription and translation. In this case, adenine will be orange, thymine and uracil will be green, guanine will be blue, and cytosine will be yellow. The first step to protein synthesis is transcription. This occurs in the nucleus. Surrounding the nucleus is the nuclear membrane. For the mRNA to be able to leave the nucleus, it must go through nuclear pores, which are these. The first step involves the helicase. The helicase goes down the DNA and unwinds the double helix. Through the use of the promoter and terminator, the RNA polymerase knows where to start creating the mRNA and where to stop. From the promoter, the RNA polymerase takes free nucleotides with complementary bases to the DNA strand from the cytoplasm. As the RNA polymerase is going down the DNA strand, the ligase is right behind it, sealing the sugar phosphate backbone. At the terminator, the RNA polymerase stops, and the new mRNA strand is free. It leaves the nucleus through one of the nuclear pores. Once transcription is over, the DNA forms back into a double helix.
The second step of protein synthesis is translation. Translation begins with the mRNA moving to the ribosome. Then, the tRNA comes down to the mRNA from the cytoplasm with an amino acid. The tRNA has a small 3-base code, known as the anticodon, so that it can correctly attach itself to a complementary 3-base code sequence on the mRNA known as the codon. The mRNA then shifts over, allowing a new tRNA to attach to it with another amino acid. To allow the amino acids to build up, they go through the process of dehydration synthesis, making methionine attached to serine. Again, the mRNA shifts over and a new form of tRNA comes down with a new amino acid, asparagine, which sounds a lot like asparagus. Dehydration synthesis occurs again and methionine and serine attach to asparagine. The second tRNA leaves the mRNA and the mRNA shifts over and new tRNA comes down again with the amino acid valine. Dehydration synthesis occurs, and all amino acids from the previous tRNA attach to valine. When the mRNA shifts this time, there are no more amino acids, and the stop codon takes place of what would be tRNA with an amino acid. The built-up amino acids break off the tRNA and leave the ribosome. This forms our new protein, known as the polypeptide chain. This protein can be used either within the cell or outside the cell.